Imagine your brain as a bustling city full of busy residents working together to keep everything running smoothly. Among these residents are the microglia, tiny but mighty immune cells that act like vigilant supervillain patrolling the nervous tissue, always on the lookout for trouble. When everything's normal, these microglia hang out in a surveying phenotype, quietly monitoring their surroundings. But sometimes they step into a more active role, becoming reactive microglia, ready to respond to injury or disease. In the case of neurodegenerative disorders like Alzheimer's or Parkinson's, these microglia can become hyperactivated or primed, reacting aggressively to accumulating debris like amyloid plaques or alpha-synuclein aggregates. They release inflammatory cytokines, reactive oxygen and nitrogen species, and toxic mediators. Think of it as a city under siege with inflammatory cytokines, reactive oxygen species causing oxidative stress and damage in nearby neurons. These reactive microglia and astrocytes can produce neurotoxic mediators, including nitric oxide synthase and matrix metalloproteinase 3, which may contribute to neuronal death through mechanisms like apoptosis, disrupted calcium homeostasis, and oxidative stress. The result, cognitive decline, memory loss, and behavioral changes as the brain's key regions, hippocampal, frontal, parietal, and temporal areas become affected. Within this neural city, dopaminergic neurons in the substantia nigra are particularly vulnerable. When these neurons are damaged by neuroinflammation and toxic aggregates like Lewy bodies containing alpha-synucleins, symptoms such as hypokinesia, rigidity, and tremors emerge, classic signs of Parkinson's disease. Cytotoxic T lymphocytes and other immune cells can further worsen the situation, causing extraneural neuromelanin to be released and fueling the inflammatory fire. On the molecular level, inflammasomes, multiprotein complexes like NLRP3 and P2X7 receptors sense danger signals such as heat shock proteins, fibroglobulins, or high mobility group blocks 1 proteins. They activate caspases like caspase 1 and caspase 3, leading to the production of inflammatory cytokines and execution of apoptosis, which can cause neuronal loss. Meanwhile, microglia communicate via signaling pathways, including Janus kinase and signal transducer, an activator of transcription, responding to cytokines and chemokines in a finely tuned balance of pro and anti-inflammatory actions. Astrocytes also get involved maintaining blood-brain barrier integrity, offering potassium ions and regulating glutamate levels. But when activated excessively, astrogliosis can become toxic, releasing nitric oxide and reactive oxygen species that further damage neurons. So in this intricate dance of immune cell signaling molecules that neurons understand, and myelinating microglia's response is key to tackling neurodegenerative diseases. After all, keeping our tiny immune surveyors in check might just help keep our brain city vibrant, healthy, and full of life.